So I think that's everyone. Welcome to tonight's program on mosquitoes. I'm Elizabeth Reinhardt with the Essex Library. Karthik Designew is the coordinator for this program, and he's going to introduce the speakers and get us started. Go ahead, Karthik.
Thank you, Elizabeth. Um, Good evening, everyone. Thank you for making it today. So today we're going to be talking about mosquito-borne illnesses, and more specifically, we're going to be talking about some recent advances in science and some recent techniques that have come up in that can help us confront mosquito-borne illnesses. So as we get started, I'd like to start off by saying that mosquitoes are very efficient killers, but how deadly are they really? So to kind of get an idea of that, here's an infographic that kind of lists the most deadly animals on the planet. And the numbers next to each of these animals just tells you how many human deaths they cause in a year. So that's right at the top, we have snakes causing about 75,000 deaths in a year. And right at the bottom, we have sharks. And to put things in context, mosquitoes turn out to be the most efficient killers, causing over 10 times more human deaths than the snakes, at about 780,000 deaths in a year. So that's an extremely efficient killer. But how exactly do mosquitoes actually go on to cause these fatal illnesses? Like, I mean, a mosquito bite is after all just that itchy patch that goes away after a while, right? Well, it turns out that mosquitoes can also carry a huge range of viruses and parasites along with them. And it is, in fact, these viruses and parasites that they carry, that they inject into you when they bite you. And that is kind of the cause of the fatal illness. And this concept will kind of be central to like following the rest of the talk as we talk about some specific solutions to um, uh, as we talk about some specific solutions to combat mosquito-borne illnesses. So now that we know the mechanism, I'd also like to introduce a couple of words that you may not have heard of in the past, and again, will help you follow the talk as we go along. So we have this mechanism that we just talked about, and clearly there are two important players here. The first, of course, is the viruses, parasites, and anything else that the mosquito carries. So these are called pathogens, and pathogens are basically the agents that cause sickness and disease. It could be a parasite, it could be a virus, it could be a fungi, it could be something else as well. And when you look at the mechanism again, there's the other obvious key player, which is the mosquito itself. So any such small organism which can carry such pathogens and spread serious diseases are what we call vectors. So mosquito is one kind of vector, but we could also have others like say sandflies or ticks, among others. So statistics say that vector-borne diseases kill about 1 million people every year, and we already saw that mosquitoes are responsible for about 80% of those. So here are some basic personal measures all of us can take on a daily basis to protect ourselves from vectors and bugs in general. The first strategy would be to try and keep them outside our homes. Using air conditioning is a great solution. Repairing holes in screens and keeping screens and windows closed, especially when there's a disease outbreak, is a good idea. The second strategy would be personal protection. Use insect repellents if you're going out late at night or if in places where there are a lot of bugs. If you find that there are a lot of bugs and mosquitoes in the area you live in, then it might be advisable to use a bed net. And finally, the third strategy would be to somehow try and stop them from breeding. It turns out that stagnant water is a great breeding ground for mosquitoes, where they can lay eggs that develop into adult mosquitoes that can cause serious diseases. So if you find that you are leaving out containers outside, just so they don't accumulate water when it rains or snows, you might want to invert them or maybe keep them closed. So those are just some basic measures all of us can take on a daily basis. But in the rest of today's talk, we'll be talking about three main solutions that science has kind of come up with. And in the first part of today's talk, we have Mara. She is a PhD student in microbiology, and she studies mosquito-borne viruses and in developing vaccine targets against them. She will present the first part of today's talk, where she will present a vaccine-based solution to confront mosquito-borne illnesses. And in the second part of today's talk, we have Sandra. Sandra is a PhD student in cell biology, and she studies mechanical stress on cells and how it promotes fibrosis. She will present a genetic engineering-based approach to combat mosquito-borne illnesses. And finally, in the third part of the talk, we have Christina, who's a Master of Public Health student at Yale, and she studies the effects of climate change on health. She will be talking about citizen science and how all of us as a community can contribute to research done by scientists to combat mosquito-borne illnesses. And she will also touch upon climate change and its impact on mosquito-borne illnesses. So with that, I'd like to hand it off to Mara for the first talk.
Thank you so much for the introduction. Oh, I still cannot share screen. Can somebody help me solve this? Okay. Okay, there we go. Thank you. Can you all see my screen well?
Yep.
Yeah, wonderful. Well, thank you, Karthik, so much for the introduction. As was said earlier, I will be talking about role of vaccines in prevention of different mosquito-borne illnesses. But to start off, here's our little outline. So we talked about personal protection against mosquitoes. Now we're going to be talking about vaccines. We will also touch a little bit on genetic engineering and on like different approaches like surveillance and citizen science. And with that, let's jump into vaccination. And before we do that, let's just talk a little bit about how vaccines work, just so that we're all on the same page. When our body needs to fight off an infection, especially when it encounters for the first time, the first line of defense is something called antibodies. Antibodies are like those things that recognize the pathogen, and then when they recognize it, they can signal to the rest of the immune system so that we can produce active response and fight it off. So when we encounter a pathogen for the first time, we don't have those antibodies yet. We need to produce them. So response is slower, we get certain disease, right? But then if we encounter the same kind of pathogen again, the response will be much faster because we have those antibodies already circulating in our system. So again, probably you will not have as much disease, as much symptomology. And we learn to replicate this more artificially with vaccines. Now, what a vaccine usually contains is some form of the pathogen of interest, let's say dead pathogen or some fragments of its surface for a body to recognize. And when it's injected, our immune cells see it as something foreign, something to be recognized and fought against, but it doesn't cause disease as a regular virus would. However, because we already learned to recognize it, when an actual active dangerous virus comes into contact with that organism, the antibodies can recognize it fairly easily and kill it. So again, no disease, no symptoms. And just to talk a little bit about how transmission occurs in general, how do we encounter those pathogens? Right, the disease transmission that we usually think about in, let's say, COVID situation, right, is when disease transmits from person to person, and in a community is going to be the cycle of transmission. However, what we usually don't think about is what about if mosquito is involved in it? It's not, most mosquito-borne illnesses are not transmitted person to person. However, mosquito bite will serve a transmission here. And this way, there's actually transmission between infected and non-infected mosquitoes and human populations. So there are two sides of this process. And if we look at an individual level, here's what happens. A mosquito carries some sort of virus. This mosquito bites a completely healthy individual and infects them when taking a blood meal because they have to like physically bite them and reach into the system. So then on this person who has virus circulating in their blood, another mosquito, completely clean, will bite, and then with a blood meal, it will take up some of the virus and become infected itself. And then again, next cycle continues, next human will get infected, and then that person will become the spreader of the virus again. And there's some unique challenges associated with this type of transmission as compared to perhaps community-based transmission. So first of all, yes, of course, we have this extra component of it, a mosquito vector, as we defined. And the problem here is that you cannot eradicate virus from a mosquito population. If we kill it off in people, there will still persist in mosquitoes constantly being reintroduced. Another problem is that virus needs to adapt to both human body and a mosquito body, which are very, very different immune systems. And it makes it very variable and very good at fighting off different threats, and just hard to target for the vaccines. And another problem, which is more of a socioeconomic perspective, is that those diseases are widely spread in developing countries, making it just hard to both develop and spread out different vaccine efforts. With that, let's get to some examples. And first example is going to be very, very positive. Yellow fever is probably not some disease you encounter on an everyday basis, but it's really widespread in a big part of the world. And at some point, a company called Sanofi Pasteur has developed this vaccine. Now, I'm going to continue referring to it as a traditional vaccine. And what I mean by that is that it stops virus where you think about it. So instead of transmission, it will stop that cycle altogether. So a person bitten by an infected mosquito would not get infected if they're vaccinated with Sanofi Pasteur yellow fever vaccine. Now, what it contains is a safe virus, exactly what we talked about when we talked about vaccines, right? So after vaccination, just one shot of this vaccine induces very efficient protection and persists for decades. And around 500 million people have already been vaccinated with this vaccine. And some people even think it is probably the best vaccine we have, like, so far against anything at all. So everything's great here. However, it's not that good for all of the viruses that mosquitoes carry, even if they're really similar to yellow fever. Let me introduce you to yellow fever's cousin, dengue. So dengue is very, very widespread and is being called the fastest spreading emerging pathogen right now, especially when it comes to vector-borne pathogens. So in 2023, the most recent data, over 5 million cases worldwide and over 5,000 dengue-related deaths. And the problem is that it's not only in the southern hemisphere nowadays, but because of climate change, it's also spreading to places where it had not been seen before. Like, for example, cases in Europe or in some southern states. And again, it has not been there before. It's not there because of travel. It's there because it's establishing itself in local mosquito populations. And just to introduce really quickly to what kind of disease it is, in mild cases, it is somewhat like a flu or a cold. However, some people can develop what's called severe dengue, which can result in hemorrhagic fever, shock, very nasty symptoms, and potentially lethality. And another problem with dengue is that it is very, very variable. So don't get too overwhelmed by this figure. What I'm just going to show here is that dengue can be separated into many, many clades. It's more variable than, for example, COVID is, and we know how variable it is. An important thing here is that in different colors, you see 4 different types of dengue. Those are what we call different types, 1, 2, 3, 4, and they're as distinct that they almost can be considered different viruses. And even when one encounters one of them, it does not form protection against another. So reinfection can occur. That's important to understand when we think about vaccines, right? Now, there has been an attempt of developing vaccine in traditional way against dengue, and it did not go exactly as planned. So Dengvaxia was developed by the same exact company as yellow fever vaccine, Sanofi Pasteur. And it passed all of the clinical trials, and it was considered safe. But it did have some side effects that it did not consider in the beginning. What was that? The following story resulted. So again, Dengvaxia is still the only licensed vaccine against dengue. 2016 was given to a cohort of school children in the Philippines. And what's happened is that some of those children have died because of dengue induced by that vaccine. Now, here's what happened. First of all, naturally, here's how the disease would go. A mosquito, continued dengue, would bite somebody. Then somebody will develop like a mild case. However, if then they encounter a mosquito with a different type of dengue, remember when we talked about different types, right? If a different type gets introduced, then they have higher possibility of severe case. That's what happens. Now, if the first person gets bitten by a dengue-containing mosquito, and then we test for presence of antibodies, and we confirm that indeed they have been introduced to dengue before. And then we vaccinate them with Dengvaxia. Then everything's great. Even if that person encounters a different type of dengue or the same one, they will not develop disease, they will not develop severe disease. That is confirmed again. However, if a person who has not been exposed to dengue, like probably most of us present here, if a person like that gets vaccinated with Dengvaxia and then gets exposed to dengue, then they have a higher chance of developing severe disease and even lethality. So what we really learn here is that viruses are not created equal. Like yellow fever and dengue, you would think it's transmitted by the same exact mosquito and it causes a little bit of like similar symptoms, but in reality, developing vaccines against them is very, very different process. and establishing different challenges. There's also some vaccines in development against dengue that are currently in phase 3 trials, meaning they are safe, they're efficient, but like it's still being proven so that it can be safely distributed. 2 of those in phase 3, first one by NIH and the second one by a company called Takeda, both protect pretty well against dengue 1 and 2. However, they do not protect well against, say, dengue 3 or 4. So maybe, just maybe, we need to look at development of vaccines against something like dengue from a little bit different perspective, not traditional perspective. Now, let's return back to our transmission circle. This here is just what traditional vaccine blocks, the way of virus entering an individual. But what if we go to block another side of the circle? What if we try to block the virus from jumping from human who's infected to a mosquito, right? Being a little bit altruistic here. But the result is that the next person will not get infected because that mosquito will not be carrying any virus. Probably not one person will be saved, but many that mosquito will bite. And this is what's called transmission blocking. And let me just show you in case of malaria, right? Natural transmission, as we used to, mosquito bites a person infected with malaria parasites. Parasites makes its way to throughout the mosquito, and then the next person will get infected with another blood meal. But what if the first person was vaccinated with a transmission blocking vaccine? Then a mosquito biting on it will take some of its blood, but it will not allow a parasite to develop. So the next person will be totally safe. And when I think about it from community perspective, this is how it would work with traditional vaccines. Some people will be protected, and some of them will not get a disease, but it still persists in a mosquito population, so we can't get rid of it. And then people who may not be able to get vaccinated for whatever reasons, they will still get infected over and over. However, we return to our original state. If transmission blocking vaccine was implemented, that'll clear a lot of virus from the mosquito population that persists there, which in turn will result in much lower cases in human communities, and also will install protection on those who may not be able to get vaccinated otherwise. But also, there's another way to think about it. What about if we protect not against the pathogen, but against the mosquito itself? And to think about that, let's zoom into the exact moment of bite, the precise moment of interaction. When mosquito bites on the skin, it has to inject parts of its saliva, just like little molecules that serve its own functions. They can aid in mosquito blood meal, but they also can be absolutely necessary for pathogen to establish itself in this new organism. And they also inhibit some of the immune response. So what if we target those proteins that are specifically absolutely required for pathogen to be present? And there is actually a first candidate for this kind of approach. It's called AGSV+. It is developed against malaria mosquitoes specifically, and it targets 4 of the salivary proteins of the mosquito. And they already went through phase 1 trials, which just means that the vaccine is safe and it does establish immune response. So next steps will be to test the clinical efficacy in larger populations. And of course, there are very, very clear benefits to this kind of approach, because if we think about virus transmission here, just imagine how many viruses one mosquito can carry, because areas in which those are popular are overlapping a lot. It can range from, our dengue, yellow fever, or Zika, which a lot of you heard about from the recent little epidemic, to very, very exotic ones like Eastern encephalitis, right? If you develop vaccines traditionally, you would have to target each one of those specifically. So somebody living in an area where a lot of those are circulating will have to be vaccinated against many of them. However, if we target just the mosquito, then we just need one vaccine. We don't need to develop 10 different ones. And this approach is really being developed also against the viruses of mosquitoes as well. So just to summarize, there isn't one perfect way to fight every single one disease carried by mosquitoes. Those can be very, very different, and each one requires its own approach that can be beneficial for broader society. And with that, thank you guys for your attention. Now that we covered personal protection, we covered vaccines, we're going to switch to genetic engineering, and then coming up, we have surveillance and citizen science. Just a quick reminder of what vaccines we talked about. And I'm gonna hand it over to Sandra.
Great, thank you, Mara. I'm just gonna go ahead and share my screen. Great, okay. Can everyone see my screen okay?
Yeah.
Yes, okay, perfect. All right, again, thank you to Mara for that great introduction in regards to the technologies that we can use to combat mosquito-borne illnesses. I'll be transitioning into a different kind of technology or solution to combat mosquito-borne illness, genetic engineering. So just a little refresh, as was previously discussed, mosquito-borne illnesses pose a unique and difficult challenge to public health. First, because the viruses that they carry have high mutation rates, making them challenging to target and to treat, as was talked about with the vaccines. And they're also vector-based diseases. So you're not just dealing with the virus, you're also dealing with the mosquito, which is a live animal and can adapt to different attempts of ours to control their population size, such as growing resistance to pesticides or insect repellents. So this has led us to be a little bit more creative in our approach to addressing mosquito-borne illness. If just reducing their size or reducing their population is difficult and targeting the viruses they carry is difficult, what if we change the mosquito itself, taking a threatening vector and making it a non-threat? And this is where this gene editing technology has come in to actually make this possible. So to better understand how gene editing works, let's take a close look at this mosquito. And I mean really close, so that we see this important little molecule called DNA. Now, DNA contains the genetic code that is unique to every individual, or in the case of this talk, every mosquito. And what this means in practice is that DNA provides an instructional guide for our bodies to make RNA. Now, this DNA to RNA conversion is the first step to expressing a gene, which I'll explain a little bit more. So what this expression means is that this RNA provides a template for ribosomes, which are the machines in our cells that make proteins, to actually construct proteins. The gene starts as DNA, ends up as a protein. And these proteins are made of different amino acids, which are these little building block molecules that we get from our environments and from our foods that make up these proteins. Now, you may hear protein and think like eggs and chicken and steak, and that is true. But in this context, proteins refer to the little building blocks that make up our entire bodies, all of us. Or in the case of mosquito, proteins are what make up the mosquito's legs, the mosquito's wings, or the little nose that it uses to bite. In simpler terms, ribosomes are the chefs, DNA serves as the recipe, amino acids are the ingredients, and proteins are the final meal. Genetic engineering allows us to change the recipe, or the DNA that we start with, and that's going to eventually change the final meal, the protein that would be expressed by that gene. So I know what you might be thinking, that is not a mosquito, that is a jellyfish, and that is true. But the reason that I'm showing it and what's special about this jellyfish is that it fluoresces green because the Acora victoria jellyfish has a gene expressing a green fluorescent protein. So scientists have been able to decode the actual DNA sequence that expresses this gene for the green fluorescent protein. In other words, we know the recipe for the green fluorescent protein. Knowing this DNA recipe, that DNA sequence can then be inserted into another animal's DNA, such as a fish. And that is how we have these green fluorescent protein expressing fish that you can actually buy at the grocery store, or not the grocery store, the pet store. So that leads us to another question. How can we edit a mosquito's DNA, something that's so small? So there are a few requirements for technology to make this possible. First, we need to be able to actually make cuts in the mosquito's DNA. If we want to take something out or if we want to put something in, we need to be able to make a cut to do that in the first place. However, you don't want to just release a pair of scissors into your DNA. Your DNA makes up the proteins, or the DNA provides a template for all of the proteins that make you up. So that could cause some problems. Therefore, we would want cuts to be very specific. So something like a pair of scissors, but with a map, knowing exactly where to cut. And what's great is we actually have this technology readily available in labs today. You may have heard of CRISPR, which won the Nobel Prize in Chemistry in 2020. Super exciting win for women in STEM. The award was given to Jennifer Doudna and Emmanuelle Charpentier. And this discovery of CRISPR-Cas is referred to as genetic scissors. And what's really cool about this is that it was discovered in bacteria, so it's something that nature actually already does already. And CRISPR stands for clustered, regularly interspaced, short palindromic repeats, which is a pretty gnarly name, but what it refers to is in bacterial DNA, scientists observed these repetitive regions of DNA with these unique DNA sequences in between called spacers. So it would be a repetitive region, a unique spacer region, a repetitive region, a unique spacer region. And while scientists were not quite sure what these regions were for, they also noticed that next to these CRISPR regions would be groups of genes coding for proteins called Cas proteins or CRISPR-associated proteins. So they noticed these repetitive sequences, these unique sequences in between, and then always nearby Cas proteins as well. So that mystery has actually been solved. We know what purpose that serves in the bacteria. Those unique spacer sequences in between the repetitive sequences in the bacteria's DNA was actually not bacterial DNA at all. It was DNA from viruses. Not only do viruses make humans sick, they also make bacteria sick. And whenever a virus would infect a bacteria, the bacteria was able to cut a little bit of the virus's DNA and insert it into this CRISPR region. And then what would happen is that viral DNA would get turned into RNA, like we talked about earlier, and it would get stuck onto that Cas protein. So now you have a Cas protein with an RNA stuck to it, originally from a virus. And then if that virus ever attacks the bacteria again, that RNA will recognize the viral DNA, guide the Cas protein to it, and cut up that virus's DNA. So very exciting, we're seeing this scissors with a map kind of effect. Whereas the Cas proteins can cut DNA, the RNA is able to guide it. So by using Cas proteins from bacteria and designing our own guide RNAs, we can make specific cuts and edits to mosquito DNA. So our next question here is, how can we actually use this technology to combat mosquito-borne illnesses? How can we apply it? And to answer this, I'll be showing you 2 case studies of how this has been done. So first, for my first case study, I wanted to introduce the Aedes aegypti mosquito. And it also is known as the yellow fever mosquito. It's one of the world's most widely distributed mosquitoes, so a lot of them everywhere. And they also carry many of the diseases that have been talked about already in this talk. Malaria, yellow fever, dengue, chikungunya, a bunch of different mosquito-borne illnesses, which makes it a problem and a good target for this genetically modified technique. So scientists have created these OX5034 genetically modified Aedes aegypti mosquitoes, and they have been created to express 2 different genes. One is a red fluorescent marker, similar to the green fluorescent protein we talked about earlier, but this one is red, and this allows scientists to be able to distinguish these mutant mosquitoes from regular mosquitoes in the wild. And they also express a self-limiting gene. And what this means is when the female mosquitoes get close to reaching reproductive age, where it's time to mate and have babies, they just die. And this isn't just to unfairly target female mosquitoes. This is because female mosquitoes are actually the only mosquitoes that bite. So in practice, what this looks like, first, these genetically modified mosquitoes in the lab lay their eggs. These genetically modified eggs are released into an area. The eggs will hatch, and the offspring genetically modified male mosquitoes mate with the wild females. Then this passes on the self-limiting gene to their offspring. So the female offspring, as they get a little bit older and almost reach reproductive age, they just die before they become adults. And the result of this is a greatly reduced population of biting female mosquitoes. So something exciting about these mutant mosquitoes is that they are approved by the Environmental Protection Agency. There are certain risk categories and rigorous testing that goes into making sure that these mosquitoes are safe. First of all, the question asked by the EPA is, do these mutant mosquitoes pose any risk to humans? It was found no, that they do not, that they are safe for humans. Is there any risk that these new genes could potentially be integrated into the local mosquitoes and give them some kind of advantage, for example, making them resistant to like pesticides or making them live longer? No risk of that. And lastly, do they pose any risk to non-target organisms? So other wildlife in those areas, other pollinators like bees or bats? No. So overall, safe for humans and safe for their environment. And very exciting, these mutant mosquitoes have already been released and used in parts of Brazil, the Cayman Islands, Panama, and India to control the Aedes aegypti population, and they've been really effective at reducing those population numbers. And they've now been approved for use in Florida and Texas counties as well. So there are some limitations of this mutant mosquito. First of all, it can't be used to control a disease outbreak that has already started because it takes a while for these mosquitoes to actually integrate into their populations and mate and die. If there's already like a big spike of a mosquito-borne illness, not going to be super helpful. So much better as a preventative measure. Additionally, these Aedes aegypti mosquitoes can only mate with other Aedes aegypti mosquitoes, which is helpful at reducing that population. But most locations have multiple types of mosquitoes that carry illness. So those will not be impacted by this mutation, and they will still be able to bite and spread illness. Additionally, when the genetically modified mosquitoes stop being released into an area, the populations then go back up and kind of stabilize. So these mosquitoes need to be continuously released. All right, and now I will move on to my second case study, focused more on engineering mosquitoes that can't spread malaria. And we did talk a little bit about the malaria transmission cycle already in Mara's talk, where we have a malaria parasite that infects a mosquito, then it infects a person, and then that person is then bitten by a mosquito, which is infected, and so on. But here's the scientists doing this project wanted to focus on the first step of the malaria transmission cycle, where the mosquito is infected by the malaria parasite. So after a mosquito drinks infected blood and is infected by the parasite needs a certain amount of time to grow and develop in the mosquito's gut. And once it's done developing, it then moves into the salivary glands of the mosquito, making it infectious and able to bite the next person. However, only 10% of mosquitoes actually live long enough for this to happen. So scientists saw an opportunity here in that delaying the malaria parasite development could potentially prevent more infection from these mosquitoes. And this is work done by the Transmission Zero team at the Imperial College London. And they genetically engineered mosquitoes to express antibacterial proteins in their gut, hopefully to delay the development of the malaria parasite. And something interesting to note is that these genes expressing these antibacterial proteins were actually originally found in African clawed frogs and honeybees. So that example we saw earlier with a sequence of DNA being taken from the jellyfish and put in the fish, same idea here. Sequences of DNA being taken from the frogs and the bees and put into these mosquitoes. So the schematic here is that a mosquito will first take a blood meal, and this actually triggers expression of these antibacterial proteins. And these antibacterial proteins then impair the malaria parasite development in the gut. It doesn't stop it completely, but by the time the malaria parasite is able to make it to the salivary gland, the natural lifespan of the mosquito has kind of ended and the mosquito is dead. So this project is in the early phases of testing. For example, they have only really tested whether this genetic modification can block transmission from parasites that have been raised in the lab. So they need to test whether the genetic modification can also block transmission from parasites that have actually infected humans. And the Team Zero transmission team is partnering with researchers in Tanzania for this, getting actual samples of parasites from infected school children, which are a really at-risk population in Tanzania for having this malaria. Yeah, in summary, genetic engineering is a powerful tool in stopping the spread of mosquito-borne illnesses. Here I presented 2 examples of how this can actually look in practice, where genetic engineering was used to reduce the mosquito population size or to create mosquitoes that cannot actually spread disease in the first place. Yeah, thank you for listening. Just a summary of where our talk is as a whole. We've talked a bit about personal measures against mosquito-borne disease, vaccination. Now we've covered genetic engineering, and I'll be passing it off to Christina to talk about citizen science approaches to combating mosquito-borne illness.
Thank you so much, Sandra, for giving me the floor. I'm very excited to continue now the presentation. I will share my screen. I hope it works. And so, yeah, this should work. Okay, so now I'm going to talk about climate change. Don't worry, just kidding. We are still talking about mosquito-borne illnesses. However, now we're going to zoom a little bit out and think of what are the changes that we are experiencing with the spread of mosquito-borne diseases and why it might be. And one of the causes, the one I'm bringing to you today, is climate change. So usually when we think of climate change or global warming, as you might have heard of it, we think of the images that we have on the back in this slide. Maybe you think of glaciers melting, or you think of the wildfires that have happened in California or in Canada or in many other parts of the world, or you think of roads closed because of floods. These are all impacts of climate change, and these all have an effect on mosquito-borne spread, emergence, and transmission of these illnesses. So yes, this is what you were thinking about when you thought of climate change, because before this talk, and hopefully after this talk, you will also acknowledge that there's so many other effects that have actually an impact on our very own lives. So climate change impacts like all of the different diseases that can be transmitted by animals, but today we are focusing mainly on mosquitoes. And since our activity today is called Science in the News, I'm bringing you some news from here we have from October, and just from the last few months, we are seeing more and more news of diseases that we thought that we didn't have in the northern parts of the world that are starting to appear. For example, in October, we got this news in the scientific journal Nature about dengue. And the news said that dengue is spreading in Europe. And how worried should we be? Well, we already learned a little bit about it and the situation with vaccines and so on. So we definitely don't want dengue to come to our latitudes. However, now that there is more travel after COVID and there's warmer summers, there have been some outbreaks in Italy and France, for example. Something similar has happened in the US with malaria. So actually in decades, for the first time in 20 years, the US has recorded some cases of local malaria. This is because now the temperature, the conditions are favorable for the mosquito. So we are starting to see these diseases appearing. And same for West Nile. West Nile virus cases are increasing in the US. Most of them are in the West and Southwest, but actually now there are rising concerns that in the East Coast, the conditions are being more favorable, and there's also a risk of this virus to come close to us. And just to think a little bit about how is this happening, like we have those news, I said that the temperature, the conditions are more favorable, but what is it that is actually happening? So first, let's briefly discuss climate change, just so that we are all in the same page. Maybe you have heard about it as global warming, because this is the most obvious effect, that the temperatures are rising. And this is because of the so-called greenhouse gases, such as carbon dioxide, methane, and nitrous oxide, that are produced from most of the industrial activities, transportation. So every time we are driving our cars, raising livestock. So most of the generating electricity, most of the activities that we are used to are generating these greenhouse gases. And what happens is that in the normal scenario of not global warming, the normal greenhouse gas effect that we need so that our planet doesn't freeze, there's some solar radiation that comes to the Earth, and it's re-radiated back and escapes into the space. And just the right amount enters back, because it's kind of trapped because of the natural greenhouse gases. However, when we are now increasing the concentration of these gases that I was talking about in the atmosphere, what happens is that more heat is trapped into the atmosphere, so temperatures are rising. And this is because the less heat escapes into the space and most is trapped back. And basically, this is our planet, this is the Earth. It's as if it had a blanket around that doesn't let the heat go. So all of these extreme weather events are happening, including the rise in temperatures. And why especially mosquitoes? Why are they affected? And it's actually not only mosquitoes. I thought it was very relevant, since we live in New Haven and we are very close to Lyme, Connecticut, to discuss that also other diseases are climate sensitive, or sensitive to the climate conditions. For example, tick-borne diseases. That's why I was mentioning Lyme, because of Lyme disease. This is a tick-borne disease that it's also highly climate sensitive. And then the mosquito-borne diseases that we were talking about today already for a long time, like dengue, malaria, West Nile fever. So the warmer temperatures, which are like these longer summers, the milder winters, as well as all these extreme weather events of floods, droughts, they're all affecting the geographic distribution. So those news about them, the diseases coming up north, the seasonality, you may have noticed that the ticks now stay longer because of this longer period of warmer temperatures for the ticks to be around, or the mosquitoes that we're discussing today. And also the incidence, the number of vectors grows because of these ideal conditions. And this is affecting indeed the diseases that they carry. And also, just to mention, there's also waterborne diseases that are highly affected by climate, such as the Vibrio bacteria. And these are the ones that, for example, when some people get food poisoning from raw oysters that are contaminated with bacteria, they're the ones that cause diarrheal diseases. And these bacteria are also more likely to survive and grow in warmer waters, that are, especially when they are contaminated waters from flooding. That is also one of these impacts that we see from climate change. And here we have some maps to show some of these effects that we were discussing with the mosquito. So as you might have learned today, Aedes is the mosquito that carries dengue. We have two different species, so Aedes aegypti and Aedes albopictus, as we've discussed today. So what we want to show in this graph is that currently this is the distribution of the mosquito and the seasonality. So the time, like the months that the mosquito is around. So if it's in blue, it means less, like 1, 2, 3. So that's what we see up north. And this means also less risk then of the diseases that they may carry. And when we get to yellow, then it would be like around half of the year. And then red means all the time. So this is the distribution currently, but under the highest emission scenario, what we mean by this is if climate change continues, so we continue burning fossil fuels at the highest rate, emitting these greenhouse gases that I was talking about at the beginning, then we might see in 2050 and in 2080 these scenarios in which, for example, if we look at the US, now it's getting, like most of the area is getting yellow and then blue and really up north to Canada as well. Same for, excuse me, same for Europe. So basically going up north, as we can see, even here, for example, in the 2080 scenario, we see that most of North America and most of Europe and Asia is at risk. And here we have, again, the graph that you have learned with Mara of the transmission of the mosquito. And I wanted to speak specifically on how does temperature affect the life cycle and the mosquito and dengue virus that is inside Aedes albopictus. I'm sorry. So the temperature, as you could tell because of this distribution, is quite crucial for the mosquito and the virus that it has inside. So the ideal temperatures for the mosquito are between 18 and 32, because below 18 degrees Celsius that we use for reference, it is too cold. So there would be no risk in this case. However, also above 32, it would get too warm. That is why maybe in some regions of the world, some of the diseases might not be present anymore. So we might see them up north, but maybe in other regions of the world in the south, they will have different diseases, or these diseases might even disappear. So it's not that we can't say that climate change will increase the spread of diseases everywhere at the same rate. It will be really different between which mosquito we're talking about, which disease we're talking about, and which part of the world we are looking at. So again, back to our temperature. Ideally, the dengue virus likes a temperature between 23 and 32 degrees Celsius to go from the midgut to the salivary glands to be ready to continue the transmission cycle that Mara was explaining earlier today, and with an ideal temperature of 28 degrees Celsius. And well, now that we know a little bit more about the risk and how it's changing the distribution and why is climate change affecting mosquito-borne diseases, what can we do about it? Well, it's not that we can do something specific about climate change, you and me as citizens, other than, of course, acknowledging that this is a problem and trying to advocate for the end of burning fossil fuels that are causing climate change and so on. But this is another strategy that we want to bring to all of you to help scientists to control mosquito populations. And this is called citizen science. So this is an example from Europe. It's an app called Mosquito Alert. So the app works like this. You can download it on your phone, and if you see a mosquito that looks like the ones that we are so worried about, here the ones that carry dengue, like the tiger and yellow fever mosquitoes, then you can take a picture with your phone, and there will be some guidelines so that you can know more or less if it can be one of those the mosquitoes that we're worrying about so much. And then there will be some kind of algorithm and technology to help shape and bring this information to the scientist, who will then validate it. And indeed, if it was correct and experts validate that this is one of the mosquitoes that we need to control, then it will appear on real time on an interactive map. This is, for example, an example from Europe. You have some of the mosquito populations reported in Spain, in Italy, and in general in the south of Europe, as that is where the temperatures are now more favorable for this mosquito, as we were discussing, because of climate change. So this is the map of the tiger mosquito. And in general, this is the cycle of what we use. So the citizen observations, collecting data, then there's the expert validation, It goes to interactive map, and then the science can be used, especially for management and control and prevention of outbreaks. And in the USA, there is a very similar app for ticks. So it would be worth, if you want to explore and check it out, to download it on your phone, the tick app. And that can also be a good tool to prevent and inform of ticks in your areas. And finally, I would like to close with the concept of One Health, because today we have been discussing different approaches to tackle these mosquito-borne diseases from different fields. And this is the essence of One Health, in which we want to address these issues from an integrated animal, human, and environmental health perspective, while working with individuals from many different fields. So Sandra, Mara, and I, we work in different fields, yet we are tackling the same issue. And indeed, this is what we need to coordinate, to collaborate, to communicate, as we're doing today, to achieve the best health outcomes for people, animals, plants, and our environment. This is the approach that also the CDC is implementing to tackle all of these diseases. And here we have as a recap. So we looked at vaccination, we looked at genetic engineering, we're looking at citizen science, and we hope that by using a holistic approach and collaborative approach under the One Health framework, we can fight against mosquito-borne disease. Thank you.
Great. Thank you, everyone. If there are any questions, the speakers will now be taking questions, and you can feel free to unmute yourself and ask away.
I had a question for, I think it was for Sandra. What about the, you talked about the gene editing for the mosquitoes? That seems like it might be kind of costly. Where would the organization doing that get their funding?
Oh, that's an interesting question. I'm just thinking about it in terms of like, so I do this kind of CRISPR and gene editing in my lab, and it's actually not something that is crazy expensive.
Okay.
It's something that we know, like making these guide RNAs, for example, like I ordered some last week and it was like $80 for like 4 of them. In terms of the actual plasmids themselves, also like, I don't know, Mara, if you've ever worked with them, but I feel like it's not too expensive.
I mean, if you think about it like that, when you already have an established lab doing it, it's not expensive. But then, of
Yeah.
course, R&D is like, it is costly. And it's mostly NIH funds a lot of this stuff. It's mostly like government funding. There are collaborations with international governments as well. Yeah, but it's mostly on the government level.
Yeah. And for something like mosquito-borne illness, like I imagine that like there's going to be government funding for that.
And then maybe I missed it, but was there a particular mosquito? I know there's different types of mosquitoes, the culver and because I know culver is kind of popular in Connecticut. Or it's populated in Connecticut, should I say. Is there a particular mosquito that you would suggest for the gene editing, or did I miss that?
Yeah, the first mosquito that I talked about was the Aedes aegypti
Okay.
mosquito, and that's the one that's just like very widespread and carries a lot of different illnesses. Which is why that gene editing was done on that mosquito.
Does anyone else have any other questions for the presenters? I think we did have a comment from the audience. Let me see if I can pull that up. Yep, this is from Stephanie. Thank you for this very informative presentation. I'm a nurse practitioner and I appreciate the update. So great job, everyone. If there aren't any other questions, then I'll just end the Zoom meeting now.
Sure. Thank you, everyone.
Okay, great.
Thanks for having us.
Have a great night.
Thank you.